Hey, everybody, this is Jason Wilson with Curious About Cannabis podcast. Thanks for tuning in again. Today, I am joined with my good friend, colleague, and mentor, Dr. Anthony Smith. Thank you so much for sitting down with me today.
Thanks, Jason. It's good to be back together talking about stuff.
The dynamic cannabis science duo.
And how.
So for those that aren't familiar with who you are and your work, do you mind just really briefly just kind of describing some of your science background and how you got into doing some of the cannabis science work that you're doing now?
Sure. Right now, working on analytical chemistry, both for safety and quality control and product development, and also in industrial manufacturing in the cannabis and hemp space. My background, I always have had a passion for human health and physiology, and I got a biology degree back in the 90s and thought, maybe I might want to do pre-med.
Yeah.
But that was just not for me. So I went to grad school at Oregon State University and became, again, super passionate about biochemistry, specifically worked on the molecular and biochemical aspects of aging in the cardiovascular system. And also mostly because my work was at the Linus Pauling Institute. Part of our mission always is to look at nutritional supplements and vitamins as they relate to, biochemistry, aging, cancer, etc. So that fit in well, with my interest in health. On top of all that, academically, I'm pretty passionate about herbal medicine. I would say like nutritional chemistry, especially as it relates to human health and performance. I've worked after grad school as a consultant with quite a few different nutraceutical manufacturers, both doing chemistry and quality control as it relates to product development, but also published research doing basic pharmacokinetics for like over-the-counter dietary supplements.
Mm-hmm. And can you explain to people what pharmacokinetics is?
Yeah, I mean, it's a big thing in drug development and drug validation. But on the dietary supplement side, there's a fair bit of this kind of work. It's looking at the level of either a compound or its metabolites as it passes through your body. And complete pharmacokinetics and pharmacodynamics, is a physical tracing of the entire mass of a dose of something that you eat. And you would account for, every milligram that you take, what your body does with it and where it comes out, essentially.
Yeah.
Basic pharmacokinetics for a dietary supplement would be maybe you take Oregon grape supplement, and then every hour or two, depending on like how we presume what happens to berberine in your body, maybe we would collect blood every hour or two for the course of a day, or maybe even 2 days, and look at the level of, in the case of Oregon grape, berberine in your bloodstream and how it goes up and back down over time. And depending on partly your genetic tone, but also the chemistry of whatever compound we're talking about, the peak area or the peak amount of that compound in your bloodstream, it might happen very fast and then go away slowly, or it might build very slowly and then peak later. Just completely depends.
Nice. Well, I know there's a lot of pharmacokinetic, pharmacodynamic work that's trying to be done now on cannabinoids to try to understand kind of how they move through the body. Because one thing that a lot of people are unaware of is that cannabinoids aren't very bioavailable in the body, primarily because they're kind of fatty, and that causes a number of issues with the body. They tend to get bound in the blood really quickly, and like basically glucose will adhere to some of those compounds, make these conjugate compounds that then just kind of get excreted. I think sometimes because, especially THC rich cannabis, because it elicits that euphoric effect or intoxicating effect, that's presumed that it's very active in the body, very bioavailable. But research is kind of showing that it's not really the case.
Yeah, absolutely. And that stems, I think, from, like the binding kinetics, if you will, of THC and its metabolites to CB1 receptors, is that very tiny amounts that make it through the blood-brain barrier are plenty enough to elicit the classic THC euphoria. I mean, I think anandamide is the typical endogenous cannabinoid that would stimulate that receptor, and a handful of molecules in your brain is more than enough
Right.
to get that
euphoria.
euphoria.
Yeah, exactly. Yeah, it's really fascinating how little it actually takes to elicit some pretty profound changes to consciousness and everything. So these days you're doing a lot of cannabis testing, and that's how you and I kind of originally came together, helping build up labs and get methods developed and validated and everything. One thing I want to make sure we spend some time on talking about is, I think, a question that's on a lot of people's minds that are in the cannabis industry is, how does a lab know that their results are accurate? Because it's pretty well known at this point that depending on what lab you go to, you can get very different results, especially with potency, and that's kind of its own issue. But even with things like pesticides and things that really affect public health and safety. So can you talk a little bit about what labs do to prove to themselves that their own results are accurate, and then stemming from that, what consumers can do to kind of evaluate labs so they can trust the results they're getting?
Sure, absolutely. It's a subject near and dear to my heart. I guess I'll maybe answer that a little bit in reverse, is that, as a consumer of what labs have to offer, if you're a cannabis or hemp farmer or manufacturer, one thing I always encourage people to pay attention to if you're shopping for labs or meeting labs is, you really should be looking for a lab that you can have as a business to business relationship with. And that would begin by, when you tour a lab, or even talk to their principals, on the telephone, like, there's no black arts in a laboratory. And so there's no like secret curtain.
Yeah.
There's honestly nothing super proprietary.
Right.
Or, there's a lot of high tech stuff. And what we do is tedious and difficult. That's why everyone doesn't do it. It's not because it's, magic or proprietary. And so, you should get that feeling when you go to a lab, like, these are our instruments. These are our trained staff. Here are some of our, records. We make data in a process. And just like if you have a factory full of robots and machines and humans, in order to get that factory output right, there has to be a lot of traceability and a lot of control and a lot of communication. And these are the same sort of things. I mean, I don't say data factory because we don't actually manufacture data, but the word for it in the lab space is that we do data realization. Okay, so data realization is not squirting stuff into machines and then issuing PDFs to people. It's a tedious process that has tons of built-in traceability and controls and checks. And when you go to a laboratory, quality should be visible. The lab should be clean. It should be organized. The people, should be wearing what you would consider typical laboratory lab coats, gloves, eye protection, things like that. So you know that they're serious about their profession. There should be training manuals posted. There should be logs, logbooks next to things like scales and instruments, right? Those are some of the early signs that there maybe is quality happening in the lab. Beyond that, what we're doing in the lab, in the back, if you will, I had mentioned traceability. So traceability is a very, very important part of quality assurance, in manufacturing, but, especially over here in doing lab work too. And what we mean by traceability is that, businesses and regulators do stuff with our data. That, our data validates that materials are okay for commerce or public consumption. Right. And so that data, when it goes out into the world, as an ISO and ORLAP accredited laboratory, we need to make sure that all of the reagents and chemicals that were in the laboratory that went into making that data happen, that they're validated and we know where we got them.
They're pure.
And that they're pure. Similarly with the humans that touch samples and computers and instruments, we have to have traceable records that the people that participated in data realization are trained to the latest procedures, that we've done what we call demonstrations of capability. They're sort of like bench checks of their precision. Similarly, that there's records that instruments are calibrated, and that, any certificate of analysis that we put out there, if any of that information is ever in question, we can do a quick audit and tell you every person who was a part of the data realization process, every batch of chemicals and standards, every instrument and what its status was. Yeah, so traceability goes the other direction too. So I just described how we understand everything that goes into our certificates of analysis. But imagine a case where one of our vendors calls us someday and, we had an accident in the lab and we shipped you this chemical or that solvent, and it was the wrong stuff or it was the wrong purity.
Yeah.
We have to now go and look forward into any data that we've realized with that material, right? So it's traceability in both directions. And that's pretty much the foundation of how you manage quality is, one, understanding everything that goes into your process and
being able to trace it back. Yeah.
Another part of quality and what you should be looking for is a lab should be doing what's called proficiency tests. There's an array of formal and informal ways to do that. We do both. Our laboratories participate in third-party external proficiency tests. So these are, chemistry and biology companies that provide benchmarked, standardized material that they will ship to a lab, and you are blinded to the quantitative amount of analytes that are in it, but this is a cannabinoid proficiency test, or this is
what to test for.
Pesticide proficiency test. Yeah. And so in the pesticide proficiency test, we'll receive ground hemp, or sometimes it's made in the state. So sometimes it's ground cannabis, and then we get a vial of liquid, which contains an unknown amount and quantity of pesticides, which we're required to spike on there, perform our assay, and then report those qualitative results back. and you either pass or fail, right? You have to get all the analytes. Generally, these studies are done with groups of labs, so you have to be typically within like one standard deviation of the actual answer. So a lab should, a lot of labs are actually required to do PT tests. But it should be something the lab is proud about. And you should definitely ask your lab about how they do their PTs and when and how they perform. That's the primary external benchmark that we have for what we call accuracy.
Yeah, you don't want to run into a lab that is nervous about sharing their participation in a PT.
Yeah, absolutely not. I don't know why you would want to. They're hard to accomplish, and it's really like the only external thing that we have.
Yeah.
We do a form of internal PTs, like between our own laboratories, but also with collegial, I guess, competitor laboratories.
Kind of a more casual round robin type of.
Exactly. We'll agree on a way or a method to produce some homogeneous material, and then like distribute it amongst our different labs, and then compare the data for our own purposes. And that's a good way to actually quantitate what we call, calculation of uncertainty. Uncertainty, it's a statistical term, and it's sort of it's roughly a balance of a measurement of your accuracy, but also precision.
Right.
And precision is something a little different, but also important. So when you just do one test on something called a PT, you're either accurate or you're not. That's a shot on target,
hit the target or
and
not.
you hit the target on the bullseye or not, or some distance from it, which is acceptable or not. Precision would be something that we generally measure in-house, and it would be a repeated preparation and measure of the same thing over and over. And it would be for the goal of measuring the precision, maybe of a trainee or a technician, but it also can be a measurement of precision of like an instrument.
Yeah.
And also just the overall process. A lot of times we'll do precision measurements by having, like I mentioned, a trainee prep 5 or 10 of the same exact thing and then measure them on the instrument and see the difference. But then we'll also have that done on different days, at different temperatures, and
get a full sense.
See what kind of variation happens, not just from the technician's thumb, but also from the instrument and the whole environment of the laboratory.
Yeah, and I imagine that can get kind of complex to calculate together, because you have uncertainty around things like pipetting. You have uncertainty around the ways the instrument injects. You have uncertainty around temperature fluctuations that affect things like density of liquids that you're working with, even variation related to sampling, which is really a big one, because if sampling is not performed, well, it kind of depends on what the purpose of the testing is, but if the purpose of the testing is to get some sort of quasi-representative value that's supposed to represent a batch of something, then the sampling error becomes a really big deal.
Yeah, absolutely. And when you're benchmarking something like pesticides or residual solvents, or even microbiology contamination, they're generally produced by what's called a spike. Like we're going to add X milligrams of pyrethrin to X grams of marijuana. And so you have this calculated theoretical, there is a known answer.
Yeah.
But when we do PTs for cannabinoids, especially what we call in matrix or actual cannabis that's ground up to be homogeneous and then sent to different labs, there's no true answer. There's not a magic machine that knows those cannabinoids, right? That's an unknown material.
It's not a perfect answer. This is 25% THC, absolutely.
Yeah, that's not known. I mean, the only way to know that would be to measure it, and well, we're all measuring it. So there is not a universal measurement to know that any more than what we do. And in those cases, the actual or presumed accurate answer, it comes out of the study. It's essentially the study mean or thereabouts.
Well, I think that's something that a lot of people don't necessarily realize is that and this is true for all sorts of analytical testing, that when you're dealing with natural products, even if you get a flower sample, grind it to a pulp where it's a powder, like sand almost, and you test that, you're still going to get some variation. And there's still some like minor amounts of heterogeneity, even in that bit. And sometimes people get really worked up, and you and I have both experienced this directly. They get really, really worked up when they expect their cannabis flowers to be some certain potency, and then it comes out different. And sometimes that difference from what they've expected is only a couple clicks, a couple percentage points off of their expectations. But because the way cannabis is being priced is oftentimes indexed on THC or now also CBD, depending on the market. Those couple of clicks can really piss some people off, and it's they don't tend to react very well to the fact that we could test this 10 different times and you're going to see some, the numbers dance around kind of a common mean or median point. And so, your flower may come out 20%. If I test it now, if I test again, it may come out 18.5. It may come out 21.5. And that doesn't mean that any of those tests were inaccurate or wrong. It's just there's this variance related to the natural heterogeneity of the natural product that's being tested.
It is, for the most part, variation in the natural product. And again, kind of going back to if you're shopping or looking for labs, this is something a lab should be able to show you too, is that, you've mentioned that there's variation in the injection precision of the instrument, in the pipetting of small volumes by technicians. And that's why we're continuously, performing repeated QC measurements like that of our technicians, of our instruments, because, when you come into the lab and I want to convince you that, you should have a relationship with our laboratory and do work here, my presumption is that just trying to impress you and say I have a fancy degree and fancy instruments is not really going to cut it. What I really want to say is that, we measure our precision, and we know what that is, and we're happy to show you how we measure it, and give you that. Sometimes it's bad news that if I test your same product over and over again over the course of a week, it probably varies in our lab as much as probably 5 or 6% difference.
Right. And that's different than percentage points.
Correct.
I want to make clear to people, because sometimes, especially in this industry, when we talk about percentages, those two ideas get conflated. So you're talking about 5 or 6% total. So if you had, if something was coming out at 200 milligrams per gram, or just make it easy, 100 milligrams per gram, you're saying that it may come out 95 milligrams per gram or 105 milligrams per gram. That's the type of variance you're looking at.
Yeah, absolutely. And then when you take that down to, THC, which is around 20%, that would vary a point or two in either direction. And that's normal precision of our process.
Yeah.
So, there's definitely factors. So that's our, say, 5% that we contribute to error. And then really like the rest is on back to the homogeneity of the material.
Yeah.
And, laboratories, the ideal sample for a laboratory is a pure granular powder that is completely homogeneous. And every time I test a bit of it, it is exactly the same.
Right.
So any variance I get in the data is the sum of variance in, again, measurement, pipetting, injection. But, cannabis is not that. It's if we're talking about cannabis flower, it's sold as a flower. Like if you test 100 milligram bit that you cut out of the stem versus 100 milligram bit you cut out of the calyx, those are going to have vastly different THC. And a laboratory, their goal or their charge, I guess, sometimes by law, is that they need to test material as it's sold.
Right.
So there's been much debate and discussion about the lab should trim the bud. And I get that that's a part of rolling a joint, but to the laboratory, it's cannabis, but it's produce.
Yeah.
Right. And it's produce in bulk. So it's, it would be misrepresentative for the lab to trim out like
all the extra leaf
and just make this really pristine sample. And I understand that sort of maybe what happens when people prepare the material for bong hits or smoking, but it's a commodity. It's a commodity produce. And we're required to basically test the material as given. That's how it goes to market.
At least here in Oregon. I know in some states, and we went through this because you and I were working together when Oregon was kind of going through some major changes in its regulations around cannabis testing and everything. But there's some states that are kind of that are roughly where Oregon was, 4 years ago or whatever, where producers are allowed to submit samples. There's actually an article that came out very recently about Washington's testing program, and a lot of they basically went through from my understanding, a pretty big audit, and found one of the issues that was found is that producers are still supplying samples to the labs rather than the labs taking the samples themselves, and so you get a lot of misrepresentative sampling that comes from that, as well as Washington hasn't mandated certain levels of auditing and QC that like Oregon has, and definitely California. California seems to be going pretty strict with testing, possibly in some regards maybe a little too far. We can talk about that later. Some of the action levels or some of the limits that they want labs to work with are a little intense. But what advice would you have for producers that live in states where the labs aren't doing the sampling? They're having to do the sampling themselves, and they want to try to have an honest representation of their batches. What kind of advice could you give them as far as trying to do the best sampling they can do to get the most representative results for their batch, rather than a sort of doctored sample?
Well, first of all, you're going to need like some real management commitment that's what they want, right? Because when there's mandatory required testing and your competitors are hand selecting samples for the purposes of getting
the highest
what
value
they want on the lab report.
Right.
That's not falsification of data, but it might be, sample
toes into manipulation,
sample manipulation, or, it's non-representative, if you will, of the bulk commodity. So, their management of whatever this company X that we're talking about really needs to be committed that that's what they want. And that's actually normal stuff in a dietary supplement and even pharmaceutical or even food manufacturing. I mean,
just basic agricultural testing. Like some of my experiences before getting into cannabis was, I was doing work with the BLM in seed collection, and we'd go visit, all of these huge farms that were collecting seed, and they'd have to get seed tested for different things, and dealing with very large batches, and they'd run into these same issues of trying to get representative sampling and testing and everything for commerce.
Right. I mean, the ketchup factory is not, sorting through their ketchup to find like the sweetest little jar. They want one that they're going to send off that's exactly like the batch.
Right.
And there is a lot of value to that in a company. There's brand stability and product quality and consistency, but there's also manufacturing and cost control benefits to really wanting homogeneous production and sampling. So assuming a manufacturer is looking to have this level of quality, there's a few different standards out there. I believe the FDA has some guidelines for sampling for natural products.
They do. The WHO does.
The
as
WHO
well.
also has guidelines for sampling. I think herbal medicines or something like that, or plant derived medicines, and they're roughly the same. I mean, it's kind of an algorithm that you must select some level of risk that you're willing to take, which would guide like how many sort of increments you would take from your batch. There's also some statistical variable for the number of containers that your batch is in. And then there's a variable for what tests you're getting, how much material the lab would actually need.
Yeah.
If they're running a battery of tests, one increment might be, 10 or 20 grams of something.
Right.
And so, yeah, I mean, I would start with guidelines like that. And I'd mentioned risk, like with herbal products, what are the risks? Is that it goes both ways, right? If you want to buy a train car full of echinacea to make a dietary supplement, there's risks to your quality control of your product. So you probably want to make sure that it doesn't have things like metals or microbial contamination, dust and filth and stuff like that. But then, on the manufacturing side, there's also some other risks that you want to manage with your sampling and testing and that probably relates to like maybe what we would call the potency or the quality of that echinacea. If you want to make a standardized product, you need to benchmark how much alkalamide content is in your echinacea bulk material before you start putting it in the bottles. So you take into account those things and then work out this matrix for sampling. It'll tell you basically how many increments and from where in the container. Maybe it's not a carload, maybe it's a 55-gallon drum or something like that. It's not super complex. Definitely can be managed, and almost no dietary supplement or nutraceutical manufacturers are forced by a regulatory body to get a third party to come and do sampling.
They just require that it's statistically sound as far as like GMP rules.
Right.
It's a very basic statement in there, but it's like, and that's why they have those guidelines from the FDA about that, and that's kind of where the what is it? 1 plus the square root of n comes from. It's one of their recommended sampling plans.
I completely understand where regulators are coming from in states where they're requiring cannabis labs to physically perform the sample collection as an independent third party. I think I understand the motivations for that, but honestly, like as a business owner and manager, it's kind of something I wish that the cannabis and hemp industry would sort of take up on their own, just like the dietary supplement and pharmaceutical industry, because it's really just a practical reason. I mean, at the laboratory, we focus on making a laboratory a business, and there's all sorts of things that we need to figure out to make that a business. Same thing in other business, cost control, training, and there's a certain level of skills and things that us scientists and chemists understand and spend a lot of time and have expertise in, and that's how to make a business of a technical operation like realizing data in a laboratory. And to add the need for that business to also have
A sampling business
a
on the
sampling
side.
business, and it's a completely separate business that's like not exactly, in a lot of chemistry and lab guys' repertoire, because it involves, logistics, scheduling and driving and maps. And so it's a weird thing to have to add to your lab business. It's not typical, I would say.
Yeah. I totally agree. It was something that when I was doing some of that work, I hated. Just
It's
as
fairly costly, and we have to pass that cost on to clients. I mean,
right, and yeah, going back to something you said of wishing that the cannabis industry, whether it be hemp or medical or recreational markets, would kind of take that upon themselves. My hope is that I can see right now regulators are super nervous in general about cannabis, the cannabis industries, going public and being legal and everything, and they're putting these really tight, in some states putting these really tight controls on them. And my hope is that after a few years, some of that will kind of calm down and start to settle into more normal, GMP, type operations. And that'll take a while because a lot of cannabis companies are going through a huge learning curve right now because they've either been operating in the black market and are trying to come into the legal market and get acquainted with all of these rules. I mean, a lot of consulting I do with clients around GMP or even like ISO accreditation for labs, this stuff is super new to them as far as just understanding what the FDA expects and understanding that, like, in Oregon, the Oregon Department of Ag, when they're looking at food production facilities and everything, they are acting on behalf of the FDA, doing that work. And the FDA contracts them out to do FDA inspections. And I think that's something that people don't realize is that your state regulatory bodies, we always think of like, well, the FDA is this thing, and then the Department of Ag for the state is this, and the FDA just contracts a lot of that work out to the state agencies, and so having really good relationships with your auditors, food safety inspectors, all that sort of thing can go a really long way. And my experience is that a lot of regulators are excited to help teach cannabis companies about these quality standards and typical business practices. They want to see businesses in their states do well and be a good example of how to do things. And so while I think it's largely a consequence of coming out of the black market or these gray markets, that there's a lot of apprehension about regulators, auditors, and that sort of thing. But I really encourage people to actually get to know your regulators and people with like the Department of Ag and everything that are going to be doing these inspections on behalf of the FDA. They can give you a lot of basically free consulting, honestly, if you just ask them questions. And
it's true. And if you leave all those stones unturned, they will get to know you.
Yeah, exactly. Yeah, you'll be interacting with them one way or another. It's much better to establish an early relationship and let them know you're trying to make an honest effort to do things as best you can. Well, let's talk about some of the things that you're seeing in the lab as far as, we've talked a little bit about potency, but let's talk about contaminants. First of all, what contaminants are you testing for broadly across all of the states and countries that you've got labs operating in? And then what are the most common contaminants you're actually finding in cannabis products?
So in general, the cannabis contaminants that we're looking at, I won't give a complete state roll call, but in Oregon, at the very least, contaminants we're looking for are pesticides in concentrates and flour, residual solvents in concentrates, and then elsewhere on top of pesticides and solvents, California, Florida, etc. we're also looking for heavy metals,
Mm-hmm.
at least the big four. Those are mercury, cadmium, lead,
Arsenic.
and arsenic.
Mm-hmm.
And then also microbiological contaminants. In various states, those would be either E. coli and or S-Tech, which is Shiga toxin E. coli.
Mm.
So it's like a sero strain of E. coli. Salmonella, Aspergillus, a number of different species required in Florida and California. And in some other states have maybe more broader, like total mold count or total aerobic plate count, which would be like non-specific bacteria.
Yeah.
And Canada has some regulations like that really just mimic kind of food and drug manufacturing safety.
Yeah. Basic screens, just like, okay,
Yeah.
there are no detectable forms of basic bacteria here, so no need to really go deeper, that type of rationale.
Classically, the most common contaminants that we find are definitely pesticides. I would say 4 or 5 years ago, it was like a rampant thing, where, at times in Oregon, some times of the year, we were seeing pesticide product failure rates at 20% or more.
Yeah.
I think the industry got a little bit of a black eye, but honestly, those rates today in Oregon, also in California for pesticide fails are, in the 2 to 4% range. It's really actually dropped. Fairly low. And I mean, back in the day, I mean, a lot of those pesticide fails were products that were, consciously sprayed on the material. Either they didn't know that those materials were persistent in the product, or didn't care, or didn't consult the list of things. I don't know.
Yeah.
But today, mostly when we talk to clients that have pesticide fails, it's either a mistake or something that was overlooked, or
sometimes unlisted,
an unlisted product, or just kind of a lack of control. And not properly trained farm technician found something maybe in the farmhouse garage instead of in the sort of validated growing area, and used it by accident or whatever. Sometimes agricultural overspray from either neighboring cannabis farms or, in Florida, a lot of product is coming up with malathion. But there's parts of Florida where helicopters fly over the neighborhoods spraying malathion, in the swampy areas. So if you're not paying attention to that in your greenhouses,
just sucking it in and blowing it over everything.
Microbial contaminants, at least on the Salmonella E. coli end, honestly, we don't really see any significant failure rates with modern, shelf cannabis. That stuff is all pretty clean. Those are contaminants, generally come from manure or bad hand washing. And so in California, occasionally we do see some problems with that in manufactured or infused products because they're, I guess didn't wash their hands making the cupcakes or whatever. Again, that's pretty low and not really a cannabis thing. We do detect Aspergillus. We use PCR or polymerase chain reaction as a screening tool. So this is a way to look for like the genetic signature of a bunch of different microbes rapidly in a reaction. And then when we do get positive hits, those are always confirmed by direct culture plates. So Aspergillus is still kind of a thing. This is why a lot of states require moisture content measurement and or water activity measurement. This is a means of benchmarking how dry your materials are and that you're cured to a certain level.
So you don't have to worry about mold or different types of fungi or bacteria growing out.
Yeah, absolutely. Also, while we're talking about mold and fungi, Aspergillus and other dark colored molds produce their own sort of form of pesticides. They're called mycotoxins. There are a number of mycotoxins that are required to be tested for, again, and not yet in Oregon, but I would imagine we'll see that within the next year or two. To the chemists, they look much like pesticides with respect to like their chemistry and how you analyze them on a mass spec. But California has a specific requirement for mycotoxin testing, and so I would say the failure rate for those are about similar to like the mold failure rate. Those are really the signature of mold.
Yeah.
But they are persistent, where you probably would only find mold in cannabis flower. Just like pesticides, we do occasionally detect mycotoxins in more finished products, in resins and infused products.
Well, there's mycotoxins once produced are super, super stable. They're very heat tolerant.
Yep.
Even under high pressure, they seem to be able to survive those processes.
And unlike most pesticides, like we know mycotoxins are hepatotoxic and persistently or cumulatively, they cause cumulative liver damage. And so like your lifetime dose, if you will, matters. A lot of pesticides, you certainly don't want a lot of them in your bloodstream. Unfortunately, you actually do from food. Yeah. But a lot of those are, very well broken down by the liver, and they're generally not that toxic below a certain level. That sounds funny to say, but still.
Yeah. Well, the dose makes the poison, right?
Yeah, exactly. Mycotoxins, not so much. Yeah, they do cause liver damage cumulatively, and so it's definitely not something that you want in your food.
Yeah, and they're pretty carcinogenic too, from what I understand. And it's something that when Oregon was developing their rules, I was surprised that mycotoxins. There was discussion about it early on, and then it kind of fell out. But it was something that we did, some work trying to look at that, and we were able to confirm like it's around for sure.
Yeah,
it's floating around. Not super common. But, just in the context of public health and safety, and especially with how popular extracts are getting, just more and more in vape pens and dabbing and all this sort of thing, I have some pretty serious concerns about exposure to mycotoxins as well as pesticides and everything. But pesticides seem to be one of the first contaminants that states catch on to that needs to be tested for. And so there's, a lot more control over pesticides than there are mycotoxins. And it's so common for producers to take flower material that might be moldy or otherwise, can't be sold as flour, and to extract it and turn it into, a concentrate and then sell that. So what about heavy metals in cannabis products?
That when states started passing rules about heavy metals, a lot of people speculated that it was not going to be a thing. The cannabis flower, there's all kinds of study that shows that it can bring or at least remove metals and such from the soil.
Right.
But if you look at those papers, not many of them actually looked at distribution of those metals in the plant, and that sort of matters. So California started requiring metals testing for all products as of January 1st. And so there's a nice little data set building there. But Florida's been doing it for quite a while, and I recently gave a talk looking at about 7,600 metals tests of various different products that we've tested in Florida over the last couple of years. And the trend is, I guess summarized that it's not very typical to find heavy metals in cannabis flower or resin. And so I think this will be borne out by some other research, but I have this hunch that, cannabis and hemp sequester metals from soil, but it probably stays in the roots.
Yeah, there was a study that came out.
I'm not promising that.
Yeah.
And I'm not a doctor.
Right. Yeah. There was a study that came out fairly recently. I can't remember exactly when, that looked at distribution and found that definitely the concentration is higher in the roots than anywhere else,
Sure.
which makes sense.
But that's not to say that we don't see heavy metal failures.
Exactly.
And
Yeah.
actually in pretty significant quantities.
Right.
And so what, I guess the summary of all of that is that the grow environment, the grow atmosphere, the grow materials, plus any manufacturing stuff downstream,
Right.
is a huge culprit in metals contamination.
Well, and something we see in the natural products industry broadly, in the context of finished products, if someone's making a manufactured product with other herbs, that sort of thing, if they're not, even if the cannabis is coming out clean, and when I say clean, I mean natural products broadly are going to have metals in them, and so it's really about the concentration, like how high is that level. It's rare to not see metals, but if you're not controlling for the metals concentration that is actually in other component ingredients that are going into a finished product, then that can cause problems, even if you've done all this control on your cannabis.
Absolutely. So as a spin out of this, this is based on real data. We're starting to help vape pen clients screen vape pen or cartridge brands. And we're actually pushing towards standardizing or validating an actual method for screening vape pen carts.
Wow.
Right? Because there's different ways that if you think there's metal in there, you can put acidic water or glycerin or vegetable oil, and for different time periods and temperatures. And so a lot of labs have started to look at this because it's sort of a big thing. I mean, I first heard of it in like 2013 or 2014. Jeff Raber of the workshop published some paper where they were finding heavy metals in the vapor stream of low quality vape cartridges. So we've been thinking about it for a while and encouraging vape pen manufacturers to, when they buy these materials, get several different samples and bring handfuls of them to the lab, and we'll wash them either with like acidic water or sometimes MCT oil.
See what might come through.
And a lot of them indeed do leach a lot of metals, and it's primarily lead.
Yeah.
But also some mercury.
Yeah.
We do see some arsenic fails at times in cannabis, and usually it comes from seems like so the story there was that I had a manufacturer or a farm, an indoor place, and they were anticipating doing metals test on their final stuff, and so they didn't really want to wait until the end and see. And so these guys were smart, and when their plants were fairly immature, just at the start of the budding stage, like we went and collected a bunch of materials. So basically immature buds, and they were loaded with arsenic.
Oh, wow.
So that, prompted a test of the soil and the water and the media and everything that we could. And turns out at the end of that story, it was a bunch of nasty rock wool.
Oh, wow.
So it was,
the initial growth medium. Wow.
It was just off the charts with arsenic.
Wow, these little things that, we get so focused on the actual cannabis product. That's so easy to not think about that. I mean, it makes sense now that you say that, but that's something I genuinely have not ever thought about, that like rock wool, might be a source of contamination like that.
But also, extractors and other manufacturers of value-added cannabis stuff, you use blenders and pumps and metal tubing to transfer materials, and glass and other containers to store those in, and those are all potentially sources of heavy metals.
Yeah. Wow. Well, I know in California they have they have pretty strict rules on heavy metals in general
Yep.
for foods and consumer products compared to a lot of states. I know that a lot of manufacturers that are trying to do production in California or trying to export to California, that's sort of a new thing for a lot of people that they're having to get their finished products tested for metals. Like in Oregon, for cannabis products, the only thing you really have to get a manufactured finished product tested for is potency.
Yep,
Um,
that's true.
and while you have to get the extract, tested for different things, it's still very cannabis product centric and not like food safety centric,
Right.
thinking more broadly about what consumers are exposed to that may not be coming from the cannabis itself.
Yeah, California requires, the finished packaged product to be tested for the entire battery. So whether you're selling
Yeah.
flour or cupcakes, those all need to be tested for all the microbiology components. If it has concentrate in it, residual solvents, and also pesticides.
What complications does that cause for the lab as far as something that I've talked to other people about before is doing that sort of testing on finished products can be fairly complicated for a lab as far as teasing out target compounds you're trying to test for, because it's in this like complex matrix of things that are you've got oil soluble components and water soluble components and mixtures in between and things that want to hold on to cannabinoids or hold on to pesticides and that sort of thing. So what sort of complications have you run into trying to ensure that finished products can be tested for all of those things?
I remember a few years ago, there was a few really odd cannabis infused products that would come into the lab that we would just, politely decline because of exactly that. How are you going to test a frozen take and bake pizza?
That's exactly the example I was thinking about.
Sure.
Yeah.
It's not, not that we didn't want to. It's just a matter of it's a big technical hurdle.
Yeah.
So, in short, mostly what a lab does is mix a sample with some solvent in an effort to leave behind the so-called sample and bring what you're looking to test for, say pesticides, into some liquid that is as clean as it can be from the stuff in the sample, but still has all of any potential contaminants. And it turns out that that's not as straightforward as it looks. And the kinetics and dynamics of how that works, for example, if you want to pull pesticides off of cannabis flower to test, when you shake that cannabis flower with a solvent and then filter that solvent, you get pretty good recovery, if you will, of all of the contaminants that are in it. Because they would just rather be in the solvent than be, loafing on the matrix, as we call it. But when you go to more complex things, chocolate, coconut oil, baked products with things like flour and sugar in them, those dynamics of, shaking the material with the solvent and filtering it and shooting it into your machine really can break down. Because like you said, extraction is about getting the target analytes off of what they're on in the sample and into something that you can look at cleanly in your instrument. And it's just not that straightforward when there's a complex mixture of things like sugar and flour and fats, or even sticky things that contain glycerin or...
And so to deal with that, are you just having to basically sacrifice a lot of, like in chromatography, you have these columns that are fairly sensitive to whatever compounds the column is not designed to take? Are you just having to go through columns more frequently than you like? Because you're kind of, as a consequence, having to inject dirtier samples than you would otherwise...
Yeah, definitely. So we go through columns quite a bit more than, like a more classical matrix lab, like an environmental lab. And then, we have lots of other procedures based on what we presume that sample matrix is,
Yeah.
chocolate or taffy or whatever, to clean up the sample, if you will, with an idea of precipitating or leaving behind some of the stuff. Gummy bears is one that's fairly challenging. There's gelatin, which is a protein. There's sugar. And it's hard to not just get a slimy mess when you extract those materials. And what we've done over time is in advance of the need for compliance testing for certain matrix types, we have looked to build a relationship with at least one manufacturer of a particular product matrix, as we call it, and in some level of collaboration, spent time validating and adjusting how we extract and clean up samples.
Yeah.
So we'll get, blank gummy bears, if you will, and then actually inject them with pesticides and things like that in known amounts, and then look at how we can recover those and get this balance of good, efficient recovery, but also, protecting your instruments from sticky messes.
Yeah. Yeah, that'll be it'll be interesting to see how other states onboard lab testing rules in the context of those finished products and the methods that get developed. Because when we were just starting work together, I mean, there was no guidance on how to handle any of that stuff. I remember spending a lot of time doing experiments with things like gummy bears and chocolate and stuff, trying to see what our recovery efficiencies were, just so for our own knowledge of like, well, how off are we when we're doing this sort of stuff, and how can we narrow that range? It's definitely more complex than I think labs are given credit for.
And sometimes it's this is why you need a lab that you can actually have a relationship with this. In the case of confections like gummy bears and hard candies, we know it's a fact that if you really put 10 milligrams of THC in a gummy bear, no matter how we work it, it's going to come out on the instrument at best about 9. That's not to say that there's not 10 in there, but there is unrecoverable cannabinoids in that material. And it's not a matter of sensitivity or anything like that. It's just a matter of like the physics and the chemistry of the stuff. There's almost nothing you can do to get that perfectly out. And so nonetheless, if you're bringing those kind of products to market, you need a lab report that says 10. And so this is simplified, but a lot of times you need to understand and have a relationship with the lab enough that they're going to actually share with you that extraction efficiency. Yeah. And you're going to have to formulate around that. If you need a C of A that says 10, you might need to formulate at 11.5. Right.
Well, and that brings up an interesting issue. From your perspective, then if that's the case, are most of the infused products on the market greater potency than what's on the label because of that?
There was no way I could surmise most.
Right. Definitely some.
Definitely some.
And it depends on the matrix.
Right. I mean, for things like coconut oil or even alcohol, ethanol-based tinctures, our recovery is 95% or better. And so those are super accurate. And not to say that gummies and confections aren't accurate, but there's no magic analyzer that can see how many THC molecules are in your gummy without completely deconstructing it. That's just how it works.
Despite some of the marketing of some of the technologies that are getting out there for like producers.
But it's true, I mean, if the lab only can get 85% recovery from gummy bears, and the lab report says 10, that product's going to be labeled and sold as 10, and it's possible that it has 11 or 12 in it.
Yeah. Well, and segueing to the next thing I wanted to get into is, from your experience seeing, in multiple states, in multiple countries, what cannabis products are testing out for, what contaminants you're finding, the potency, all these different things. From your perspective, and this is a grossly oversimplified question, and I know you can't answer it simply, but from your perspective, are most cannabis products these days in regulated environments safe to consume? Ignoring things like um, issues with THC and people taking too high of a dose and that sort of thing, but just from a contaminant perspective.
I would say in regulated states in general, I feel pretty confident that most products you get are safe in the sense that they're, probably don't have pesticides and solvents. They probably are fairly consistent in their dose.
Mm-hmm.
I mentioned vape pen cartridges. I mean, in California, we're required to actually extract the material out of finished vape pen cartridges. So if those have metals,
you'll see it.
We will see it. But in other states, you would test the concentrate before it's filled into cartridges. So
you'll never get that data.
My personal choice is, I'm not personally very interested in things like vape pens, really because of that.
Mm-hmm. Yeah.
But other than that, yeah, I do believe that in regulated states, their products are safer and more consistent than ever before.
Yeah. Well, let's change gears a little bit away from testing and talk a little bit about one concept that I want to talk about is molecular signaling. So totally switching gears here, more into your molecular biology background. One thing I've been asking different scientists that I've talked to is to try to describe to a lay audience what molecular signaling is, how to conceptualize it, and a lot of times when people are exposed to molecular signaling, the lock and key analogy is used. And you and I and with others have discussed how that analogy is really inaccurate, and that there's a lot more complexity going on with molecular signaling than the idea of you just have, let's say, THC and it fits into, this THC sort of shaped, hole in a receptor and it causes these effects. So can you describe with a lay audience in mind how to think about molecular signaling and how the body processes compounds in general and some of those dynamics that happen at the receptor level?
Well, the lock and key is a good analogy to start with. I mean, the concept is that cells on their surface have locks, if you will. So these are proteins with, different shaped pockets that are designed to sense things in their environment. In the body, most of those are from in the body.
Mm-hmm.
So, cells that line the bloodstream sense all sorts of chemistry and things and hormones but also nutrients and vitamins and such that are floating past it, if you will, but also the proteins and locks, if you will, our surface receptors within your tissues all are also reacting to what we call the extracellular matrix, right?
Mm-hmm.
And this is kind of how cancer is controlled, where a bone cell expects its neighbor to be a bone cell and to identify itself as a bone cell through signaling. And if something in there is not a bone cell, well, there's like a pretty violent reaction, whether that's an infection or a cancerous cell. But the lock and key model does come up short in that, and you alluded to this, is that there's, with a lock and key, there's one key. And it will make the lock work. And most signals and signal transduction cascades aren't that simple. There's usually a number of different keys that would fit in, and some of them would elicit either high or low bias response from that receptor, or Some of them would say turn the receptor off, while the others would stimulate it. And so it's much more dynamic in that sense. What we do know is that most receptor types, at least in broad classes, are known what class of key, if you will, they will bind to. And then also underneath the surface of the cell, what happens in what we call signal transduction is that when a receptor is stimulated or blocked, on the inner surface of that receptor, it is connected to all of the biochemistry of the cell. And so there are pathways that are turned on. The stimulation of a receptor from the outside might turn on, certain compounds that migrate to the cell nucleus to turn off and on RNA and gene expression. The receptors might also control things like, one of the ways cells control sort of their electrical tone, if you will, is by calcium and other ions being pushed to either side of the membrane, so they can actually maintain an electrical potential from their outside and inside. And so these are controlled by how pumps in the cell will pump sodium outside of the cell, and preferentially potassium inside the cell. Similar with calcium and magnesium pumps. Those things are all controlled by signal transduction. A lot of, some of the signal when you stimulate or block a receptor end up suppressing certain cell effects, and others turn on cell effects. One of the main ways that cells can sort of stimulate biochemistry to happen is that there are enzymes that are floating in the cell or ready to do work, but they're inactive until they bind to a calcium ion, right? So there's a way to have these enzymes made and ready to go in the cell, and then when the enzyme needs to get turned on, the cell will release calcium ions, and those calcium ions will sort of now bind into that enzyme and now turn it on.
Wow, yeah.
Right, so almost no extracellular receptors that are tied to intracellular actions do one thing.
Yeah.
There's almost always like a symphony of things, and sort of like the analogy that I don't know how it fits with the lock and key, but imagine, if you hear tones, we can describe their note and their volume and such. And so you could imagine that you could have a system that was controlled by tones. Like when this note is played, this happens, and when this note is played, this happens, right? But then, how does your model work if I give you a chord instead of a note? And that's an analogy of what's happening in signal transduction in a cell. So, when a receptor is turned on or off, there is generally a cascade of several different things that happen. And then this is also dramatically different in the cell type. Right, so some signals will stimulate metabolism and gene production, say in hepatic cells or liver cells, but they might completely suppress that type of activity in a cardiac myocyte.
Interesting, yeah.
Right, so the pathways are all, I don't want to say generic, but similarly mapped out is how the chemistry and electronics work. And they're definitely electronic. This is sort of microcoulomb electronics, but cells are definitely electronic, and so is their biochemistry. An example is like a hormone, right? Like a prolactin will cause a certain response in your smooth muscle cells compared to in central nervous system tissue. And cannabinoids are much the same, there's the classic CB1 and CB2 receptor. We know something about how cannabinoids bind to them. Typically, it's a binding kinetic measurement is sort of like how tightly it fits in. Like,
Those the chi values.
yeah,
Ki.
exactly. And some receptors will bind very, very tightly with a ligand, or, in this case, what's binding to it. Some bind very softly. Some of these signals, the key, if you will, can be antagonistic, which means they completely turn off and block that receptor and keep it from being turned on by positive signals. Some of them turn on that receptor. And then there's also receptor tone, where if you, and the analogy here is in your nose, is like if you repeatedly smell a strong smell, this is a cyclic AMP dependent signal transduction in your nasal epithelium, where for whatever reason, when receptors are hyperstimulated, the receptors themselves tend to either get turned off or removed.
Right. Well, I know this
from the cell.
I know this happens with a lot of receptor types, but with CB1 receptors, if you're using THC-rich cannabis a lot and you're bombarding your CB1 receptors, eventually they tend to retreat into the cell, and then the cell basically recognizes that, oh, that's something we need to break apart, get rid of.
Right.
And so over time, you can deplete the concentration of CB1 receptors in your brain and other tissues.
And these are, these signal transduction elements in your body, they don't exist
in isolation.
They don't exist in isolation, and they're not just there, to be manipulated by their environment.
Right.
And that's why this is a natural homeostasis. If you get a signal that turns on something in a cell, you wouldn't want that to be turned on permanently.
Right.
And you wouldn't want it to be turned up at a gain that's too high. And so all cells are always modulating their tone in this fashion.
Yeah. Well, and it's really interesting to hear you talk about how the same receptor in different cell types or tissue types will do different things. And it highlights how careful we need to be in interpreting certain forms of cannabis research that, for instance, like cell culture studies, or, just exposing a cannabinoid to a certain receptor in isolation and not specifying, not really looking at any other dynamics other than just really basic affinity.
Right.
You can understand the affinity that a cannabinoid or other compound, there are a lot of compounds now that we're finding have affinity for cannabinoid receptors, like flavonoids, certain terpenes and everything that, just because we understand the affinity doesn't mean we necessarily understand what they're doing in the body.
Absolutely. A lot of stuff is like that, and cannabinoids very likely fit into that same paradigm. Vitamin D is kind of an example of this, where, vitamin D's job is to put calcium into tissues.
Yeah.
So, if you have calcium in your blood, then in the presence, and this is very simplified, but when there's vitamin D present, that's a signal cascade for the blood vessel epithelium to absorb that calcium out of the blood and to put it into the cells. But if you don't have calcium in your blood, say from dietary intake, your body has a perfectly good reservoir of calcium in its bones. And so vitamin D without dietary calcium can potentially cause bones to release calcium out of the bone and into the bloodstream. And a little bit of that is perfectly fine. It's, again, part of our homeostasis. But, this is an example of how vitamin D can cause your blood and muscle cells to absorb calcium, but they can cause your bone to break down bone and release calcium.
Wow. Yeah.
Same signal.
So, kind of linking this to, we were talking about some of the limitations of cannabis research in the context of the complexities of molecular signaling, and how there's a lot more going on in the body than sometimes is made apparent in how research is communicated to the public. And you described that the same receptor in different cell types and in the context of different tissues and physiological systems will do very different things. You mentioned, sort of dose response and how receptors do different things depending on, how much of a compound is binding and what compound's binding and all of that. What advice would you have for people that are, they're going on Google Scholar, looking up cannabis research papers, trying to, interpret findings? What advice would you have for people, especially a lay audience, in reviewing research publications that talk about pharmacology and these effects, and what some of the limitations are, and what should they be thinking about when they're reading those papers?
Well, one thing, and this goes right back to, thinking about signal transduction, is that a lot of science papers are either built on or in some cases supported by what we call cell culture research.
Yeah.
Usually the studies, unless they're like straight pharmacology, aren't completely about cultured cells. And so cultured cells, what I mean by that is that we can get bits of tissue from different animals, humans included, and essentially break them apart from their tissue mass and grow them in flat-bottomed bottles with synthetic blood, if you will.
Yeah.
And so there's all kinds of different mammalian cell lines that we call them, that are used for all sorts of research that relate to drugs, disease, environmental control, detoxification, etc. And these are very important models. But I think when you see research, if it's built on or even supported by cell culture experiments, keep that in mind, right? So often, if it's important enough, a study will use several different cell line types and look for, some theme in whatever metabolism they're looking for in those models. Sometimes you'll see clinical work, so like actual, physiology or, clinical outcomes that maybe produce a theory that it relates to a particular tissue and
Right.
and signal. And so they will try to support that hypothesis by testing that signal again in a cell culture model. But the thing about cell culture models is, until you in your research mind or your research repertoire really built up a large library, if you will, of understanding many different cell culture lines, it's not a good idea, I think, to make lay conclusions that are based on a single observation of a cell culture line. It's not really happening with cannabis and CB1 receptors yet on this level, but if you look at some other classical, drugs, or sometimes even natural products, ibuprofen, or I mentioned berberine early. You can Google berberine and you can see what berberine does in dozens of different mammalian cell lines. And I think until you really can sort of digest that host of data and think about it scientifically, it's not a good idea to make scientific conclusions or assumptions about any one of them.
Well, and I think there's a lot of issues right now in sort of clinical cannabis work, that people are reading papers and drawing conclusions either from cell culture models or rodent models and saying like, okay, we understand what, these cannabinoids are doing in the presence of certain receptors. We understand how it's going to affect the cell or this tissue, and thus, we can extrapolate that out to medical conditions and start to make assumptions about what types of chemotypes of cannabis are going to be particularly effective for different medical conditions. And while, maybe eventually the science will get to that point, I don't think it's there yet. I know you've seen some of the charts that are out there that list, all these cannabinoids or all these terpenes and then all these different pharmacological effects and then little checks, what does what. And basically, usually not outright saying, but implying that a consumer can look at that and say, oh, I want to support bone mass, so I'm looking for, CBD, or I want to, try to control munchies, so I need to look for THCV, these different pieces that are typically born out of either cell culture models or rodent models, not human trials. And that being said, there have been some human trials on certain conditions, but not many. And so one thing I wanted to get across to listeners is those charts are really, really misleading because of the lack of context that's presented with them. And they're in like every dispensary that you go into.
It's true. If you have a broken arm or you're searching for a cannabis with this or that cannabinoid isn't necessarily going to support bone health.
Right.
I mean, bone health is sort of like a point of your body takes care of its own bone health. You can't really turn that on or off. I mean, unless you're like nutritionally starved, it does it in the way that your genetics have been presented to you more than any cannabis could affect.
Yeah.
The one thing that comes to mind is like if I were to blindfold you, Jason, and take you down to the symphony and pull off the blindfold maybe just for 10 or 15 seconds and then put it back on, and I would ask you to, what is this? And, if it was Beethoven's Fifth, you would say, it's, the heart, the most important thing about the symphony is, it's the kettle drum. Or if I did it at a different time, you would say like the oboe.
Right. Or that piano.
What classical music is all about.
Yeah.
Because, those are some notes or some chords, but it's really a symphony.
Yeah. Yeah. Well, I want to start getting around to wrapping this interview up. I appreciate you being willing to give me so much of your time. One thing I wanted to ask you, just to deviate away from cannabis for just a second, what's something non-cannabis related that as a scientist has you excited these days that you're paying attention to, interested in?
Oh, I like that question. Thank you. These days, something scientific that I'm becoming super passionate about, learning and I guess experimenting in some fashion as well, is brain EEG. Sort of in the context of, I'm interested certainly in how cannabis might affect that, but just more broadly, fascinated with how humans are going to leverage this technology of like being able to read electrical signals from the central nervous system, and I'm super fascinated with modalities that present it as some sort of feedback to you. So neurofeedback is definitely a part of EEG work. There's a number of really high quality home user EEG instruments that have come out recently. Quite a few of them have software with API interfaces that you could develop
like apps on your phone,
applications and stuff. And so I'm definitely interested in the science and technology and software there. And then I'm fascinated with how we can assimilate it. It's doesn't make sense just obviously to like see graphed waves. There's not much that can help you do, but there's some interesting ways that these can be presented back to a human in forms of feedback that are salient, that somehow could maybe drive psychology and personality growth and development, and I'm definitely interested in that sort of the esoteric end of kind of what that means to human personality and psychology.
Yeah, what we can actually control if we have that feedback.
Yeah. So I'm interested in mind and brain development and experimentation.
Yeah, totally.
With technology primarily.
Yeah. So one other question I wanted to ask you is, and we'll try to keep it brief because we've been going a little over an hour now, but what are two of the biggest misconceptions that you run into regularly that people have about either cannabis broadly or cannabis testing?
One of the biggest misconceptions that I think still persists is that that there's a particular strain of cannabis that you should take for a particular indication. Like you should smoke the Blue Dream if you have a sore neck, but you should smoke the Blue G, the OG, for diarrhea, and you should smoke the this or that for a twisted ankle.
Yeah, well, I think some of what we discussed in this interview is makes that, I think, pretty clear why that's problematic.
Yeah, I mean, I think research may eventually bear this true or not, that there's a few axes of perception, I would say, in the so-called entourage effect. And then beyond that, probably most cannabis strains in your body are, it's fairly mysterious how they might differ from each other.
Right. There's an assumption that these days that it's all about terpenes that provide those nuanced differences, but we don't really know that.
Yeah, certainly not, because if you eat a terpene mix, it doesn't seem to give you the same effect as like smoking or vaping it. Yeah. I think the terpene contribution to entourage effect physiologically is entirely within the response that we refer to as aromatherapy.
Yeah.
Right? Aromatherapy is not hokum. Like when you smell aromas or combinations of them, they affect your nervous tone, which can definitely affect sort of your immune tone, and also your mood. And you're changing your mood definitely affects blood chemistry and immune tone. And so I don't think it's hokum at all, but I think the sort of the homeopathic level from a dose perspective that you get from just smelling aromas probably contributes a lot more to the entourage effect than some magic thing that happens. Like, I think if you eat different cannabis strains and they all go into your gut, you don't see the differences that you might report if you vaped or smoked them.
Yeah, and something that's important to tease out there too is with aromatherapy, it's been found that, if you get a whole host of people together and you have them smell different smells, they're going to react differently to the same smell depending on prior experiences they've had with that smell. A common example is lavender. It's usually assumed that the smell of lavender is soothing and helps people go to sleep. But if you grew up having a negative association with the smell of lavender, that is not the effect that smelling lavender is likely going to have on your experience. And so with terpenes and aromatherapy and that sort of thing, it can be very individualized and what those responses will be, which then creates greater limitations to being able to say, oh, if you just target this terpene profile, you're likely going to experience this effect, particularly when it comes to things like mood and like psychological effects.
The other one is probably not as important, and I would say maybe the indica sativa thing. That it's not actually a thing,
right? Yeah,
anymore. I mean,
yeah,
it might have been in the 70s when there were, the family tree of cannabis. We could still see that it came from two branches. There's so much hybridization in modern cannabis. It's all hybrid.
Yeah. Well, anyway, I can hear my daughter screaming in the background, so go ahead and get this wrapped up. But thank you so much for carving out some time so we can hang out together and talk. I hope we can get together again soon. We have plenty more to talk about, so I hope we can do this again and get into some other interesting topics. We've only really scraped the surface of a lot of things. So
absolutely.
Anyway, thank you for sitting down with me, and I'll catch up with you again soon.
Thanks, Jason. I appreciate getting together and chatting and look forward to more of it.
Cool. Sounds good. All right. Thank you for listening. If you want to find out more about the Curious About Cannabis podcast, you can go to cacpodcast.com or connect with us on social media on Facebook, Twitter, or Instagram, or check out the Curious About Cannabis YouTube channel, where we're posting video clips from some of these interviews, as well as other educational content about cannabis science and other cannabis topics. Thanks so much for listening, and I'll talk to you later. Thanks, and take it easy. Bye.